So let's talk about Everdzi. Can you tell me the story behind the drug and if there were any initial challenges in its development?
Of course. So the idea behind Everdzi was to develop a new medicine to treat people with spinal muscular atrophy, which is still and was at that time the commonest genetic cause of infant death. So it's a genetically driven disease that is caused by the loss of function due to mutation in a gene that leads to the absence of a protein called SMNP. And the opportunity we saw was that to use a backup gene called SMN2 that produces the same protein inefficiently, and to improve that efficiency by utilizing a technique called splicing. So essentially looking at the mRNA that comes out of the SMN2, modifying its splicing into a form which was stable and therefore generated normal protein. And as you can see here, just by this story, it's a little bit science fiction-y because we're essentially manipulating the genetic machinery of our normal cells to restore a lost function. So the basic initial challenge was just purely a scientific one. How do you do that? How do you do that very specifically, but also very efficient and very safely as well? And so that was really the story behind this, was to try to provide an alternative therapeutic for these patients that corrected the defect in a safe and effective way. At the same time, because other alternatives, other technologies used, we wanted to develop an oral, essentially something that could be taken very conveniently at home, that did not involve any sort of specific procedure, so essentially would reach the maximum number of patients with this condition out there. So there were numerous challenges, as you can expect, because there's a scientific challenge on one hand, finding the right medicine, finding the right molecule, and then developing it quickly and fast enough, and also engaging with the broadest range of patients possible to ensure that the most of the benefit would come out as soon as possible.
Okay, so you touched on splicing modulation in your response before, but can you provide a little bit more detail about how it works?
Sure. So what happens for any of our genes is that the gene is translated into a long molecule, messenger RNA. That messenger RNA is composed of elements which are then transcribed into protein and elements which are not, right? There are silent pieces of mRNA. Those silent pieces need to be edited out of the mRNA before it's translated. And that process is called splicing. Right? Now, by doing that, it's actually a really core biological process. By doing that, from a single gene, our cells can extract, if you want, multiple different versions of this of that product, if you want, and so multiple different proteins. So that's a very, very conserved process. What happens in SMA is that this backup gene, SMN2, has a mutation in it, so it has an error in it, if you want, that missing of the mRNA message inefficient. So instead of generating normal SMN protein, it generates this deficient form that's missing a key component, and what's called the exon 7 component. And that protein missing that exon 7 is degraded very, very fast. So what we did was to essentially restore normal splicing to this SMN2 gene splicing process, and therefore generate normal protein.
Okay. And why was this approval such a significant milestone for patients with SMA and for Roche as well?
Well, I think it is a major milestone for multiple reasons. And the first time, it's the first ever development of an oral small molecule medicine splicing, right? There are other technologies, as I mentioned, that can be used to do that, but this was it is a scientific milestone in that sense. The first time this has ever been done. Right. I think therefore it's a great scientific milestone. The second thing, I think it's a great milestone because it's the first oral medicine that these patients can take. And therefore that has, implications for how convenient it is, how many patients can access it. Just to give you one idea, the medicine was launched during the COVID pandemic. So a lot of patients were at home. They couldn't go to the hospital. Having a medicine at home that you can take easily and safely is obviously a huge advantage. So for the patient community, I think it's a major milestone because all of a sudden they have another option, which is very safe, efficacious, convenient, can be taken at home, And as I mentioned, we developed it not for a subset of patients, but for the whole patient community, because SMN are as young as neonates and as old as 30, 40 years old, right? So everybody has access to this medicine. So that's a major milestone. And for Roche, I think it is a major milestone because it just speaks to our commitment to rare diseases on one side, but also to really doing creative science, to doing breakthrough science on behalf of patients, right? So something to be proud of.
Okay. And the splicing modulation platform, what influence do you think it will have on the future of drug development?
Well, I think when you look at RNA splicing specifically, it's one of those core biological processes, as I mentioned, that if one can modulate it, if one can learn how to tweak it to benefit to therapeutic purposes, could have tremendous potential impact, not just in SMA, but across a range of diseases, right? Of course, everyone's trying to do it in different ways, and as I mentioned, there's different ways to do that. If we learn how to do that efficiently and safely, I think it'll open up opportunities to create new therapeutics for a range of conditions, from rare ones to much more common ones. And therefore, there are a number of companies out there who are working on this, and we're one of them as well.
Okay, so the next question is about small molecules.
Yeah.
They're a well-established drug format. What benefits do they have in terms of mRNA targeting? And also, what advantages do you think they have over other drug formats like advanced medicines and biologics for treating disease?
Well, if you typically for splicing modification, right? And I think one of the advantages of small molecules, clearly, it's you know, as I mentioned, they're usually orally administered and absorbed. And therefore, they have a convenience aspect which supersedes other therapeutics, which are injected or have, you know, they need procedures to be administered, like spinal taps or IV infusions, etc. So that is a big element of this, right? That convenience, which you know, it's not just a lifestyle thing. It actually allows more patients to have access to medicines in a way that's for their lifestyle. Yeah. The second aspect, which I think is important, is the fact that small molecules are a technology, if you want, that we've known for decades now. And they're relatively, once you know what you want to do, it's relatively cheap and accessible. So that means that the costs of therapy are usually less than the cost of, let's say, a gene or another technology that does that, right? So again, it allows us to make these medicines available for a much bigger swath of patients all across the globe, right? And therefore, access becomes much more, you know, equitable for the large majority of these patients. And I'd say thirdly is that molecules, you know, because they're all orally administered usually, they reach the whole body, right? They're systemically available. So they go into all the tissues, basically, more than others, but all the tissues, right? And I think that's super important, especially for a condition like a genetic condition, where we know the genetic defect is essentially in all the cells of our body. And therefore, that was also something that we felt might be important for these patients, was to have a medicine that reached not just the central nervous system, but also all the tissues, like the muscles and the joints and the lungs and the heart, et cetera, because we're basically correcting a genetic defect, which is common to all cells.
Okay, and then the next question is about mRNA targeting. Obviously, this area will expand over time. What lessons do you still think need to be learned as this field develops?
Well, I think the main thing is that the secret, if you want, is if you use small molecules, is really around finding molecules that are very specific. Because again, mRNA splicing is a core process that is conserved. And therefore, if you're not specific, the danger is you're going to start modulating the splicing of other genes as well. And that might have safety problems associated with that. So finding very, very specific molecules, because that usually means they're very safe molecules. And secondly, finding molecules that have the good properties as drugs, meaning that they're absorbed well, they're metabolized well, they don't accumulate in other parts, you know, so all those things are almost like problems that you need to solve target by target, so gene by gene, I'd say. of course, what Everdzi has done is shown that that is possible, that feat is achievable. We can do that. And I think because we've possible together with our colleagues at PTC Therapeutics to develop the platform in the first place, now we're in a much better position to start understanding what molecule characteristics best fit this splicing modification, right? So it will accelerate the development, I think, of other new splicing for other conditions.
Okay, and how did you feel when you heard that Everdzi had won the Drug Discovery of the Year award?
Well, I felt very, very proud because, it is on one hand, as I said, for us as a company, a demonstration of what we do best, if you want, which is really following create medical breakthroughs. I felt very, very proud because for patients worldwide, I think this is going to be something very important for them, and we're already seeing that. We're seeing how much of an impact it's having in so many people out there. And thirdly as well, because I think I felt very, very proud because it is a scientific breakthrough. It is one of the things I think important for the future in terms of drug development and creating new medicines, right? So I was just really proud of the whole team that has listened for years on this, both at Roche and PTC and the SMA. So this is really, I think, one of those great examples of collaboration that shows us at our best.
And then just as a closing remark, do you have anything to say about small molecules and why this is still such an exciting area for companies to be involved in?
Yes, I'd say we're living through a very interesting time, I think, in terms of drug development. We have so many different tools and platforms to use now, right? All the way from small molecules to large biologicals to antisense and gene editing, and it's just great to have so many platforms, so many tools, if you want, so many technologies that can help you develop medicines. And I think that small molecules historically are the oldest ones, offer certain advantages that others don't. As I mentioned, they're usually simple to manufacture and therefore much more easily available for the majority of folks, right? We've learned through chemistry throughout the past 150 years of chemistry research how to be really, really good at manipulating small molecules and creating the right drug characteristics, right? So I'd say they're always going to be a core pillar of drug development. It's hard to underemphasize how important they are for so many people out there, right? The other ones are very important as well. And we get very excited when we hear about the successes of targeted medicines like monoclonals and things like that. But small molecule and core, let's say, core chemical research, I think is still a major, one of the major things, one of the major pillars of technology we need to rely on to develop new medicines.
Okay, and then just finally, is there anything else that you'd like to add, perhaps that we've missed in the conversation, or just general points about Roche and its drug development pipeline?
Yeah, I mean, maybe again, I'll just mention that, as a company actually turned 125 years last year, our whole history is really based around this concept that if you do great science and you follow the science, you create great medicines, and society and the public out there will reward you for that, right? So that makes it sustainable. And I think Roche is really a great example of that, because that commitment to science and innovation and doing the right things does pay off. And therefore, for me, Everdzi is just a great story that exemplifies what we do at our best, right? It's really about using, in this case, as you may argue, an old technological platform to do something really, really breakthrough that wasn't thought possible. To do that because we had a clear vision of what we wanted to achieve in medicine, what was the outcome we were hoping for, how many patients we wanted to be able to help, and to which extent, and then sticking to that vision through all the challenges that come, because scientific breakthroughs are still a major challenge and we still fail more often than we succeed. So when we succeed, it's that much more sweet, because then, all the effort is made worthwhile. And now that we're seeing Everdzi being launched all over the world and being accessed by thousands of patients worldwide is just incredible to see the outcome of that, the benefits you can say, and we haven't stopped. The future continues, and right now, beyond the original indications that we have, we're actually trials now in neonates that have no symptoms in the prevention mode to really try and give these babies as much of a normal life as possible. So I think this is still very much the beginning of the story for this